For your cordial welcome and this introduction. At the outset, I would like to thank the organizers and ICTS for this opportunity, where I'll get to share our experience in building a map, and we have also reduced the map into a smaller network, and then we have looked into some of the modules and the kind of system level responses that the map elicit and the smaller network provides and the modules provide. So we'll see. So as we progress, the first part of the talk will focus on the general ideas and systems perspective, and in the second part, I'll introduce you to the map that we have developed for metastatic breast cancer, and the third part of the work will explore the assembled map through various analysis procedures. We'll discuss the insights, and then we'll have a discussion. So the broader questions with which we begin any biological study is how cells or organisms do what they do, or how cells perceives and interprets the information that is there externally or internally, and then how cells make critical decisions. So the simplest answer would be central dogma proposed by Professor Crick. The blueprint is there in the DNA, and then it gets translated into protein, and then we see the phenotype. So simply we can say that phenotype is the function of genotype. However, this function is not a linear function, it's a highly non-linear function. So emerging data suggests that it is layers of network, and then multiple level interactions that's happening at each of this level is influencing the phenotypic characteristics, with lots of information transfer happening, information fed back and fed forward across all these multiple layers. So again, at any given instance, cells perceives multiple different signals, and it has to decide either to grow or stall or move, express gene, or induce death pathway, and so on. So how does this perceiving the environment, or perceiving the internal milieu and analysis decisions happens? This all happens at network level, at each individual cell and at the organ level. So this is a snapshot of a fission E cell cycle interactome, where we see only gene and protein interaction, and then this is the human metabolic network, which I had taken from Stanford University's website, which is used for their biochemistry course regularly. So without this name, we would misunderstand this to be a railway map, or cities, a water distribution map or something. So so this is our own map of TGF-beta induced epithelial mesenchymal transition in metastatic breast cancer. So such complexity requires versatile tools such as mathematical modeling, and there are people who have been thinking about networks and then network level responses, and then pioneers like Professor Lee Hartwell also has suggested that quantitative predictions and detailed simulations of these biochemical processes are necessary to develop simplifying and also higher level models and finding general principles. And the fundamental structural function property of the networks are also crucial to understanding the design space of life and to get deeper understanding into the governing principles. And people have been asking, are there special laws that apply to biological systems? And it seems they are doing all these processes so effortlessly. However, there are so many things which are happening underneath these networks, which are resulting in the functions that we observe. So no network is an island into itself. So they're not isolated units or static units. They're all functional units. So if we just take a look at these two systems, phenotypically we can see that both can fly. The natural system is could fly because of its evolution and its design and so on, and the engineered system was inspired by this natural system, and it has gone through a lot of fine tuning and lot of design, and now every module and every little part is well documented, and we engineers know where to look for and where to troubleshoot and so on. So if we see the emergence of complexity of biological system, right from the DNA's 4-letter code, which can give rise to infinite possibilities, and then the transcription, which again the 4-letter combination, which can give rise to numerous possibilities, and then many different protein combinations that is possible, which results in the interacting network. So it is the billions of years of evolution which has fine-tuned at each of this level, gives rise to the units of proteins, which again interacts in different ways to observe the phenotype. So if we could understand how the individual parts of the system works in the biological systems or in natural systems to yield the end phenotypic characteristics, we can engineer the biological systems to cater to the human needs in a better way. So while discussing about complexity and the emerging ideas, in many of his talks, Professor Paul Nurse, who has received Nobel Prize in Medicine, along with Professor Timothy Hunt and Professor Lee Hartwell, says that one of the emerging area of this century is biological organization, how the networks themselves are organized. So the organization at molecular level, at cellular level, and at the tissue level or at the physiological level, all of these are crucial. And then why is it necessary to make sense of vast pool of data that we have, and to understand the key decision making, and also to extract the information that is encoded in these interaction networks. So how is it done? It's done with the help of science, and then some of computations, and then mathematical modeling, and computers are used for solving these things, and so on. So with this, we'll move into cancer systems biology. We know that cancer is characterized by uncontrolled growth and proliferation. It's heterogeneous, and there are many acquired capabilities which are documented as hallmarks. And one of the hallmarks that we are interested in is this activation of invasion and metastasis, and this allows for cells to tumor cells to invade the other organs through intravation and then extravation. So there are many different pathways which are influencing this process, and the pathway that we are interested in is TGF-beta signaling pathway, which again promotes the epithelial to mesenchymal transition through series of interactions, and then this allows for cells to acquiring this mesenchymal behavior or partial EM phenotypes or quasi-EM phenotypes, which is helpful in this spread. So among the various signaling pathways, which we will be focusing is on TGF-beta signaling pathway. So the approach that we followed is we start with this question or hypothesis, and how EMT transition occurs when TGF-beta pathway is induced, or when there is excessive TGF-beta that's available. And then we looked for models, and then we also explore some of the experimental data, and then we go about assembling this molecular interaction maps, and we started building some small models, and then the network kept on expanding, and some of these models or small modules were modeled, and then some of the insights taken in, and so on. So the network grew, and then we had different versions of models and so on. So for network assembly, we have used so we have done an elaborate literature survey, and we have used tools like Cell Designer, YED, and then Minerva to curate the net. So curation is by literature survey, and then to draw the network or to develop the network. So all of the components that we have seen is, and it is drawn by adhering to systems biology graphical notation. It is also SBML compliant, which means we can import this and use it in other tools for our analysis purposes. So this is also annotated for the Minerva platform. So we can get to see its username, linked publications in the PubMed, and then in the Uniport and so on. So the network is imported into a cytoscape, and then the graph's properties were analyzed, or the graph's topological properties were analyzed, and then some of the plugins of cytoscape such as CytoHub and then ISMAX, which is a subgraph finding tool, was used to identify the subgraph and then the core hub regulators. And then, the discrete dynamic modeling or logical modeling was utilized to study the behavior of the network, where each of the interaction is represented as a simple logic. For example, if we look into this B here, so when will B get synthesized? Whenever A is not there, and B will get synthesized, and if B is expressed, that we call that we depict as 1, and if it's not expressed, that's 0. So here B gets synthesized when A is not there, and if we come back to C, so C would get synthesized or would get expressed when A is not there and B is available. So B and not A. So, and we have used for updating scheme, we have used both the asynchronous, we have explored both synchronous and asynchronous update scheme, and tools like Cascue, which if we provide the SBML file, it automatically generates the logical rules if we had drawn according to the SBML complaints. So, and then this is simulated by using cell collective, and then smaller modules are simulated by using Boolean net. And then for some of the modules, we have also used continuous dynamic modeling framework, where we consider synthesis, degradation, or accumulation, or multi-protein complex formation, or enzyme dependent activation. And for this, we have used mass action kinetics, Michaelis-Menten kinetics. If there are multimer formations, we have also used Hill equation, cooperative behavior observed. Then we have also used the nonlinear Hill equation, which captures the sensitivity or ultra sensitivity which is embedded. So as I mentioned, we were interested in seeing TGF beta signaling and its contribution for breast cancer development or in metastasis, because it has this opposite role. So it also in the beginning it functions like a repressor for proliferation, or it does not promote proliferation, but then later on it accumulates and then supports proliferation. And then it also is observed to be overexpressed in many of the breast cancer cells and tissues, and it has almost crosstalk with all of the major signaling pathway. Yeah, we can do that. So for smaller modules, we have done that. So for the whole network, so we have used this tool Cascue. So for a large network, it's really difficult to get to each component and writing by ourselves. So we use this tool which reads the interactions and it develops its own tool. So we will see some of the results, and yeah, it considers all the other logical rules. It's an openware which we can experiment. So the map which we have assembled has about 312 distinct species. So about 160 proteins, 30 genes, 48 RNAs, and 54 complexes. And then so all the proteins were depicted using this rectangle. So all the SBML notations were used. RNAs in this rhombus structure, receptors, gene, and activation is in black line, and inhibition with this red line, and then the blunt arrow indicates inhibition. And we can see that just by observing we can't understand anything about the system unless somebody introduces it to us. And it's also difficult to write our own code for such large systems. So here we have our, all the cell surface receptors, TGF-beta 1, 2, okay. So then, here we have canonical signaling pathway and then non-canonical signaling pathway. We also see that, we really can't, distinguish between canonical and non-canonical signaling pathway like, classical cell biology or, biochemists used to do. So there is lots of communication happening and lots of interconnectivity that we see. And here are, all the RNAs which are associated, and here we see the EMT phenotype. Now everything is, coming down towards the EMT, and then, here are the EMT regulators and transcription factors which are converging at this EMT. So I'll walk you through. So how do we read this? So the shaded part, will disappear. We will zoom into the bright part. So here we see that TGF-beta receptor 1 and TGF-beta receptor 2 complex which are getting activated, and then this is activating these SMAD dependent pathways over here and SMAD independent pathway over here, FAC and FOXQ and SMAD which are getting activated, we can see over here. So this is again the right side where we have seen the receptor. So here we see that there are signaling going to for example E-cadherin and so which is an important marker for epithelial phenotype. And here so here we see that so here we see HER2 receptor and then other Wnt beta-catenin receptors and EGFR and Notch and interleukin 6. So to some extent we have also tried to capture the crosstalk that's happening between TGF-beta induced epithelial mesenchymal signaling and also the other receptor, other crosstalks and so on. So here we see that there are activation and inactivation going on, and looking into the SMAD dependent signaling, here we see that SMAD complex is forming, and I forgot to mention, so we have considered two compartments, both the whole cell and cytoplasm, and then here is the nucleus. So the SMAD complex that is formed and phosphorylated is shuttled into the nucleus over here, and SMAD independent signaling, here we see that AKT which is getting activated, and then STAT3 and CMYC and so on. So here are the RNAs. So here we can see that MIR 200, MIR 34, and also regulators like SOX4 which are interacting with these miRNAs, and there are signaling which are conveyed all the way to the transcription factors. And if we zoom in over here, we see the beta-catenin and E-cadherin, and there are these signaling which are converging into epithelial mesenchymal phenotype, which is the phenotype that we are interested in seeing. So this map is uploaded into Minerva platform. So where so you can access this map, and you can search, and then the associated papers you will see, and also the UniProt IDs and ensemble IDs, and if there is Hugo names which are available, so those are also linked to the map. So you can access, and then you can pick your favorite molecule, and then you can see both ways, and you can track how it is leading to the phenotype, or how it's getting initiated and so on. So so anyway, this map is a static unit. So what's the point of developing this map? So we wanted to see the whole response of this map. That is also a kind of a validation. If this works, it should result in the absence of TGF-beta. It should not get into mesenchymal phenotype, and in the presence of TGF-beta, it should transition into the mesenchymal phenotype. So so the map was converted into the logical codes by using CASQ, and then it is simulated using cell collector, and asynchronous updation scheme was used. And here we see that so these are the initial conditions, and there is no TGF-beta signal here. And what we see here is only the epithelial markers, or epithelial epithelial phenotype associated regulators. So what happens when we give TGF-beta signaling? So presence of TGF-beta input. So we can see that epithelial markers are starting to fall, and then there is this rise in mesenchymal markers, and so various different mesenchymal markers are are in the rise. So the interface is where this epithelial mesenchymal hybrid phenotype or quasi phenotype is observed, the zone where it is marked. And so we have moved into analyzing the network properties. It's kind of the static property. So this is a dynamic property, this is a static property. So how well connected is the network? So we have analyzed this as a directed network, which means that we have preserved the signs that each of the edge carries, whether it is an activator or inactivator and so on. And here the undirected graph just accounts that if there is an X and Y, these are connected to one another. So this again gives some information about the network topology, and it's a well connected network. And then we looked into the most influential regulators, and it's an arbitrary number. I don't have a reason why I had chosen 25. It's an arbitrary number. So we wanted to see the top 25 regulators. So we have used this tool, CytoHubba, and so depending on this color code, this is most influential regulator, and then but here the least influential, which means maybe here at 25th ranking. So this is one of the largest clicks in terms of network biology terms, or this is the hub regulator that we have seen. And here we see that P53, RAS, and also twist and CMYC and P21 cell cycle inhibitor, and cyclin D activator of promoter of cell cycle, and P38 and E-cadherin. So all of these regulators, which are really crucial for promoting epithelial mesenchymal phenotype, or EM hybrid or quasi-EM phenotypes, are observed over here. So for those 25 regulators, we have gone in and we have looked at the Jeffia database for their significance in overall survival and also in disease-free survival. And some of these regulators, about 11 regulators, were highly significant with p-value less than 0.05. And for these, we have looked into the TNM plot to see, how they are getting, regulated in the normal state, and also in the tumor state, and also in metastatic state. So here, we can see that all of these, 11 regulators, including PAC2 and also snail, and then EGFR receptor, P53. So all of these are highly significant, and this again tells us that from a static network we can do small dynamic simulations to see whether it captures the physics of the system or physics of the network. And we can also see what are the crucial regulators, and how crucial they are, and so on. And then in our lab we have also been working on other maps for non-small cell lung cancer, and then colorectal cancer, and hepatocellular carcinoma. So we wanted to see what are the top regulators, and here again it's an arbitrary number that we have tried. So in all of these 4 different maps that we have developed, or which are in progress. So the top 16 regulators that we see in each of this network is we can see that EMT, the phenotype itself, because that is also considered as a node. And then we see snail, beta-catenin, and then CMYC and P53. So here the mesenchymal regulators, we can see epithelial regulators, oncogenes and proto-oncogenes, and cell cycle associated regulators are seen only in colorectal cancer. So we don't know the reason as to why cell cycle regulators are picked over here, but P53, CMYC, E-cadherin, snail, and all we see across all these 4 different maps. So then we asked are there any repeatedly occurring pattern in these larger networks? And we see that this kind of a motif or a module in graph theory terms subgraph where X directly activates Z some regulator, and then it activates a regulator through an intermediary step. It uses other regulator. So this kind of a structure is known as feed forward loop or coherent feed forward loop if all the signs are positive. So if all of these edges are activators. So in breast cancer, we have observed that 26 such coherent feed forward loops. In lung cancer, 22 feed forward loops, and colorectal cancer 19, and hepatocellular cancer 32 such feed forward loop structure. So if they are repeatedly, so the argument that people put forth is that if there is a subgraph which is frequently selected, then it might be contributing for some function. And so this is something which is well known in bacterial transcription network. We wanted to go ahead and see that. And one of the limitations with this kind of tool or this kind of analysis is that we get to see the numbers, but then we really can't go back and pick which regulator. So we just roughly know these many feed forward loops are there. So this tool does not provide us with such information. We are still working on this. So how to extract the actual coherent feed forward loop. So in the meantime, we have zoomed in, we have looked into the network, and so these are some of the hub regulator associated feed forward loops, and also we have feedback loops and toggle switches. We won't get into toggle switches today. And then we have built a reduced model. So a reduced, two different reduced model. So we see that this kind of coherent feed forward structure. So this is only for SMAD dependent signaling. And here we see that SMAD dependent signaling also has a feed forward loop here, and then there is another feed forward loop like structure over here, and we wanted to go ahead and analyze this. So at the same time, we have also read this paper by Adler and Metzito, where they talk about interacting subgraphs or interacting network motifs. And then we have seen that so these network motifs are interacting. So there is one node which is common between these two different coherent feed forward loops. So we wanted to analyze them individually, and we also wanted to see what kind of response does it provide when we are analyzing them in this in this architecture where it is layered. So it is two coherent type 1 feed forward loops in this layer. So this is how it's gene regulation. We know if we are if we want to think in terms of gene regulation. So there is TGF beta, which is stimulating SMAD, and SMAD complexes in P53 activating MDM2, which is directly interacting with and activating SNAIL. So we have for this we have written our own code, and then we have used this Boolean net tool for writing our own codes, and we have analyzed this in both and OR logic framework. When we are analyzing it in the AND logic, it requires both SMAD signaling and also MDM to be present for SNAIL to get activated. So here we see that there is a delayed activation, and here we see that in the OR logic it's sufficient to have one of these stimulus, so it goes on for some more time its activity, and then it falls off when the stimulus is removed. So here the stimulus is removed. So this module was also modeled by ODE based modeling or chemical kinetics based modeling, where we see that when there is this stimulus SX that is present, so SMAD is present, and then MDM2 is present. So stimulus for these two are present. So MDM2 is starting to get accumulated immediately. However, SNAIL starts to get accumulated after some time. And then we have also seen how would it respond for a brief input signal. So these are pulses of signal, which means that it's not experiencing continuous stimulus. It's just experiencing pulses of stimulus. Here the other stimulus for MDM2 is kept as 0. So then the brief signals are inducing the intermediary regulator MDM2. However, it's not reflected in activation of snail. So it filters out, this kind of brief signals or brief inputs. So this kind of network architecture, and then we wanted to see how it will respond if we introduce couple of pulses of SY. Here that's kept as 0. So here when we started introducing it, so this coincidence of both the signal was sensed, and then this is transferred to snail. However, it was kept as brief, so we do not really see snail getting activated, and only when it is persistent it's getting activated. So and then it is this similar structure, but the regulators are different, and then we observe similar property of noise filtering, and then sign sensitive delay element. So the expression of snail is delay, and then we have N-cadherin, which has similar network motif, and similar behavior is what we observe. And then we wanted to see how when these two modules are coupled together, how is this response. So there is a temporal delay that we observe over here, and then so for this kind of coherent type 1 feed-forward loops, temporal delay is known, and it's been documented for E. coli transcription networks and yeast transcription networks and so on. And we also observe that this also filters out noise, and so if we see the signal is kind of filtered out, and then only when there is a definitive signal there is this snail which is getting activated. So brief stimulus is not activating epithelial mesenchymal transition through snail or through E-cadherin. So when we increase the Hill coefficient, so we used to represent nonlinear Hill coefficient. When we increase Hill coefficient, we get much sharper graphs. So it's just graph. So here we have another interesting architecture. So here we have coherent type 1 feed-forward loop, and here we have coherent type 3 feed-forward loop, where snail along with Zeb represses E-cadherin, and then here we have seen that it is promoting N-cadherin, which means promoting N-cadherin is promoting mesenchymal transition, and simultaneously, so these are all simultaneously happening, though here we are studying separately. Here E-cadherin is repressed, and so this is the logical model for this E-cadherin which is available is repressed, and this is the ODE based dynamic modeling that we see. And so we see that this kind of reduced model, and then looking at smaller modules tells us what kind of functions they might be involved in. So so where is it leading us to? So it kinds of tells us, so we are discussing somewhere above this double negative feedback loop that which is well characterized between snail, M34, miRNA 34, and then miRNA 200, and then N-cadherin, E-cadherin, and so on. So we are looking at the layers above this. So the layers above this, and where is it leading us to, and are there similar studies? So there are similar studies that are carried out in budding yeast cell cycle regulation, or in pheromone sensing. So here we can see this network. So this is a dual negative feedback loop, SIC1 and cyclin B, cyclin B5 and cyclin B6, which is promoting this G1S transition. So SIC1 has to be repressed, and CLB5 and 6 has to be upregulated for cell to move from G1 to S phenotype. And if there are pheromones that is present, cell has to get arrested. So the pheromone signal is transferred through a linear pathway, and then there is this kind of a coherent type 1 feed-forward loop above this switch. So what these two papers from Professor Scotheim's group is telling us is that above this double negative feedback loop, there is this a coherent type 1 feed-forward loop which is channelizing, kind of channelizing the signal, that is coming through pheromone, and then, they say that switch-like transitions insulate network motifs to modularize biological network. So it is not going to affect this switch. However, it gets filtered out. So also, what their other earlier experiments suggest is that, at high level, or when FOR1 is accumulating fast, it is this. So when FOR1 is accumulating fast, it allows for rapid cell cycle arrest and then, re-entry. So it is both, promoting cell cycle arrest and then, re-entry and then, progression into the subsequent phase. So here is, the switch, like the switch, toggle switch that we see. So if we go back to this network, so here also, we have these toggle switches, and then, we observe these coherent and, see, we observe this, hypermotifs. So when two motifs are, linked together, the term that is used to refer to them is hypermotifs. So where, this hypermotifs, so it is this hypermotif, and then, the other hypermotif with, N-cadherine, which are kind of, influencing this, bistable kind of a response. So above the toggle switch, there is this coherent feed-forward loops which are influencing these decisions. And when it is synthesized at, very less, then, it sort of, impedes G1S transitions and so on. And they also say that motifs are insulated by these switches. However, it is not, affecting the core cell cycle process, which is majorly driven by these regulators, for which, this toggle switch is very, very crucial, which means that they are not, these coherent feed-forward loops and, in this hypermotif like architecture are not, interfering with the core cellular behavior. And that's what I think, we also see here in the TGF-beta-induced epithelial mesenchymal transition. So I would like to say that, the whole is greater than the sum of its part, agreed. So we have seen the whole, and we also wanted to see the simpler modules and how the simpler modules are functioning, and the simpler modules are giving us clues into how the signaling is transferred, and how it gets filtered, how it gets that time that is needed for its accumulation, and then how it is supporting the downstream decision. So if we know the modules, it can help us in solving the larger puzzle. So with that, I would like to summarize this work. So we began with just looking into signaling pathways which are influencing cancer and cancer metastasis, and then we have when we started putting these smaller puzzles together. So this has led us to TGF-beta induced epithelial mesenchymal transition, and then we have seen many of the, so the map also captures many of the crucial regulators, and they are in these motif and toggle switch feedback architectures, which is again during our analysis have seen that are very crucial for filtering the noisy inputs or brief stimuluses, and for persistence detection and coincidence detection and transformation of these informations. And again these modules are interacting in these complex network to provide some interesting information transfer, and then it helps in understanding cellular decision making. And it also says that people have been arguing that in epithelial mesenchymal transition the intermediary state and reversibility of it, and how when it reaches when it is reaching its niche in metastasis, how it is initiating this process. So I think it is these kind of coherent feed-forward loops and a hypermotive kind of architecture gives this flexibility to reverse these bistable or multi-stable decisions that the cell has to take, or the tissue has to take. So this is true for cells, this is also true for cluster of cells. And with this, I would like to thank the scholar who has taken in this small idea, and then who had grown it, and who had developed this idea and then who has done all this work. And I would like to thank my mentors, former mentors during my postgraduate studies, who had introduced me to mathematical modeling in terms of enzyme kinetics, or doing small experiments on monads kinetics and predicting that those kinetics. And then I would like to thank my mentors, Professor Sharad Bhartia and Professor K.V. Venkatesh at IIT Bombay, who had introduced me to the systems biology, or core systems biology, where I worked on modeling cell cycle regulation of fission yeast. And then, so while we were presenting this map, one of the earlier version of this map, in a disease maps community meeting, it's Professor Ananiakis who has suggested using CASQ to check whether the map is capturing the biological properties. So CASQ and cell collector was suggested by Professor Ananiakis. And for developing the map into Minerva platform, Professor Marek has helped us, and for uploading it into the website, and also to deal with this website related things, Karan Lamore and Rohit Rahman of Medflex India and Professor, it's startup of Professor KVB. So they have helped us with uploading the map into a Minerva platform. And I would like to thank NIT Warangal and its seed grant, which has provided with a good workstation, which is supporting, this kind of large networks and large network level simulations. And then, Bhavani would like to acknowledge NIT scholarship and facility. And so lots of these ideas, or very, very simple, and then work was possible because of students, and the questions students ask, and encouragements of friends and colleagues at both NIT Warangal, where I now work, and my former workplace Shastra. So with that, I would like to thank you all for your attention, and if there are any questions, I would be happy to take questions. So we begin by looking at couple of pathways which are there, and then we start we literally start to put together the puzzle, and it's an iterative process. We don't rely on one paper or couple of papers. We have to keep cross checking, and then even after assembling the map, almost after assembling the map, we get to read some papers. It's an iterative process. We always go back. And so we look at the cell line based work, and we also look at so large number of these comes from these cell line based work, where they have used breast cancer cell lines, different types of breast cancer cell lines. So that is one quality check that we can keep to maintain the robustness per se, or quality. I don't know if I can call it as robust. Not immediately, because it needs many rounds of cross-checking when we put together and redrawing and all. I don't think AI can help, but AI can help in seeing the regulators. It can give me the clue, like it can give these are the regulators. It can generate a network. Then if we want specific pathways, if you want to put the specific activation, inactivation, and then feedback and all, we have to go back to the literature. So AI could be an initial step with which we can begin. Yeah. So that might give us the list of regulators, and then some connections, and then even when we look into these databases like string, which gives it gives the list of regulators, and to some extent the interactions. So if we want to know specific roles that they play, and activation or inactivation, we have to go back to literature. As far as this map is concerned, we have left out the not so sure kind of arrows and repression activation. So whatever for the regulations for which we had convincing evidences were only put into this map. So it's not in no way it's a complete map. So it's also an evolving map as data comes, or as we read more, and we might have missed something also. So as data comes in, as we encounter the data, we have to keep updating. So the emergent property that we see is, this temporal regulation, and then, this noise filtering, and also the persistent, filtering the noise. So these are the emergent properties. And then, if we put in, the core regulators, that had been explored. So what sort of emergent property would come out? We also don't know. We are working on it. Yeah, so what I'm thinking, or so it's just, a thought experiment. So it would tell us when, epithelial and mesenchymal transitions are dynamically, perhaps, we can see, this switch working. No, we have not looked into that. However, it's possible to do. So that's in plan. Maybe in the future we'll try. Yeah, it's all, the map is for same cancer type. Not same cell line, breast cancer, various different breast cancer cell lines. So the data comes from various different breast cancer cell lines. That, we don't really know. We are just, looking generically into breast cancer. So whether, we have to make adjustment to cell line specific map is we are not sure. That's a good question. So maybe, if it is HER2 positive, we can keep the HER2, and if it is HER2 negative, we can keep it to 0 if we are going for logical model, and that could explain, those kind of. No, those kind of things is not there. Conflict. Conflicting data. No, conflicting is, so far, we have not, encountered conflicting data. Yeah, that. So far, we have not looked into other direction. It's actually, the literature that came from the budding East is sort of motivating us to think into the other direction and how the whole works. So the other direction we have not looked into. So maybe, but we can try. We have everything there. We can try